Good afternoon. I'm very, very pleased to introduce our lecturer for today, Dr. Palevsky. He is a researcher in the Department of Entomology at the Neve Yaar Research Center, ARO, since 1919. He did his undergraduate and graduate studies at the Hebrew University of Jerusalem, graduating in 1997, and his postdoc was in Oregon, USA, on IPM and orchards, and at the Hebrew University on spider mite control in date palms in southern Arava desert of Israel. He established and served as a convener of International Organization of the Biological Control Integrated Agriculture, Akorali Biological I'm jumping. Excuse me. Working group from 2007 to 2017. Wow, Eric, a long time. His current research on soil food web is focused on the utilization of soil amendments, and here we are getting closer to our talk today, for the conservation of beneficial organisms. Can I ask? Yeah, you muted me as well. So the areas that Eric is interested in are agricultural archaeology, biological control, soil predatory mites, and soil food web interactions. So Eric, really thank you for willing to give this talk today. And if you could please introduce the topic of your talk, it'll be great. Thank you. Eric, are you here? I think you're muted as well. Eric, you're muted automatically.
Okay. Thanks, Lior, for the introduction. So the topic of my talk is black soldier fly frass as an amendment for conservation, biological control, soil and plant health, and ultimately regenerative farming. I added that because perhaps we will also have a part of the audience belonging to the regenerative farming community. And my co-authors are from Humboldt University, Diana Rueda Ramirez and Professor Lidiana Rus, and Shira Fadech from the Neve Yaar. And in addition, I'd like to indicate my present collaborators on this topic: Lisa Kornblum from Machon Volcani, and Lior Shotiel from Tzafon, just right now, and Christina Cruz from the Faculty of Sciences of the University of Lisbon. So I'm going to give a I'm not going to start with a black soldier fly frass. I'm going to end with it. I'm going to build up towards that. And the idea is I want to impress upon you a holistic approach pertaining to conservation, biological control, conservation agricultural, and so on. And this has taken me many years to realize, and I want to share these experiences with you. So I'm going to talk first about conservation, biological control above and below ground, and how this pertains to regenerative agriculture and conservation agriculture. And then towards the end of the talk, I will talk about utilization of black soldier fly frass as a biofertilizer for plant protection, the promotion of plant biomass and yield, and ultimately soil health. So what is conservation biological control? The aim of conservation biological control is to protect crops against pests by promoting and conserving biocontrol agents naturally occurring in the environment. Towards this aim, naturally occurring vegetation or planted, such as hedges or cover crops, can be utilized to provide pollen, nectar, prey, and microhabitats needed for the conservation of biocontrol agents. And my field, I am an acarologist, which means that I work on acari. Acari is a group of organisms within the class of spiders. It's a subclass in Arachnida, and my focus for many years has been the utilization of predatory mites for the control of pests. So many species of predatory mites feed on a diverse diet, making them excellent candidates for conservation biological control. And these alternative food sources can conserve and even augment biocontrol agents, thereby sustaining a standing army of predators for improved pest control. So I've worked, most of my career, I worked on above ground biological control. And one of the ways that we did, or one of the methods that we used was to provide alternative food for biocontrol agents. And one way of doing this is providing pollen. So we need predators to feed on pests, but predators don't usually have pests. They need to survive in the environment, and one of their alternative foods is pollen. And so we've provisioned pollen in different ways. Other researchers have also done this, and by providing pollen, you can enhance predator populations, and then when the pest comes along, the populations are sufficient for them to control the pest. So this is an example. We worked with the engineering department of the faculty of the engineering department of Mechon Volcani, and they provided us with a pollen applicator, and we was an electrostatic sprayer. We applied first water and then applied pollen. Here you see the pollen that sticks to these drops. And then these predators, which are only 0.5 millimeter long, will feed on these pollen grains, and then their populations will go up, and they'll be more abundant. And then when the pest comes along, they are able to provide protection. Now, how is this done? In a more holistic way, natural way. You can use cover crops, and they can release pollen, and this pollen is blown onto the leaves of trees, and then this enhances the populations of predators. So this is an experimental setup that we used. We had rows of cover crops, rows of Rhodes grass adjacent to the avocado, distant from the avocado, and we showed that rows that are adjacent to the rows that are adjacent to the cover crop have higher predators than rows distant from the cover crop, and they also have lower pest populations. So this is an example of how you can manipulate the system, and basically the idea is to restore the biodiversity to enhance the biodiversity of an agricultural system. And this biodiversity basically allows predators to be more abundant and to protect your crop. So this is a way of doing this above soil. At the time, I thought it was a very good system. And now with years, I think that we can actually improve the system by not using a grass that was an exotic species, but actually learning more to use components that are endemic to the system. And the idea is to bring back nature, or to bring back biodiversity into your agricultural system. It makes it more robust, and makes basically allows your predators to function better. So this is a way that we've done this above soil. And how can we do this below soil? And here, obviously, it is more complicated. And I'll show you some slides to give you an idea of how complex below systems are compared to above systems. So soil organisms are a very important part of the ecosystem. Here you can get an idea of it in different ecosystems. You can see that if we look at all the fauna and we look at the soil fauna, so these are the two last bars, you can see that in most systems, the soil fauna comprises most of the fauna that there is. So basically, the soil is like a jungle under our feet, and the diversity is just mind-boggling. And this is an idea of the different the different trophic levels in the soil. So we have macroarthropods, and we have here we look at the size. So macrofauna, mesofauna, and microfauna and microorganisms. So the mesofauna includes mites and small insects, and the microfauna includes one cellular organisms and nematodes. And this is just to give an idea of the diversity of mites. When we talk about mites, this is a huge group, and I work mostly on this group here, on mesostigmata that contain the predatory mites and the prostigmata that contain most of the pest mites. But mites are also ticks, for instance, and there are water mites. And a very important group within the soil are oribatid mites. And these, for instance, include mites that are also parasitic to animals and mites that are pests of storage, and these also include dust mites. So this gives you an idea of the diversity of organisms, of groups of organisms in the soil, and so we have free-living nematodes. These are beneficial organisms that can feed on bacteria. They can feed on fungi. There are also a small percentage of nematodes also feed directly. They're plant parasitic. We have obviously what we called before these soil engineers. So earthworms. We have mites that feed on nematodes, mites that feed on mites, mites that feed on, we have insects and mites that feed on insects. So it's really a zoo. And so what approach can we use to support these soil food web interactions, which will in turn contribute to the health of the plant and its biomass and its yield. So yes, mites are very abundant, and as I said, there are many groups of mites within the soil. Most of them are in the upper layers of the soil, but there are also mites that go further into the soil. And it's very important to understand the biodiversity of these mites and how our practices affect their ecology and their ability to protect our plants. So these are some stunning images that I took along with a very talented group of researchers in Beltsville, Maryland, at the USDA campus. And what you see here is a mite, a predatory mite that's feeding on. It's about to feed on a free-living nematode. Here it recognizes the nematode with its front leg, and then it catches the nematode with its. It actually holds the nematode between its palps, and then with its chelicera, it cuts into the nematode. So here we see the good guys eating the good guys, and you're going to ask yourself, well, why is this important? And why should the good guys be eating the good guys? Well, as you'll see further on, there are a lot more nematodes than there are mites in the soil. They're very, very abundant, and they have many roles in soil health, and they are indicators regarding the health of the soil. For instance, if you have soil that most of the nematodes you're finding are plant parasitic nematodes, this is already an indication that your soil is not healthy. Whereas if 90% of your nematodes of the soil are free living, then these are indicators that the soil is healthy. And then when you say free living nematodes, there are many functional groups. There are nematodes that are predators, there are nematodes that feed on insects, or nematodes that feed on fungi, nematodes feed on bacteria, and so on and so forth. And this is just to show you how these predators feed on nematodes. So nematodes, you can think of them as like a bag, sort of like like children drink this chocolate drink from a plastic bag. Well, here obviously simplified, but the idea is that the mite feeds on the nematode. It basically ingests all the liquid within the nematode's body, and then it spits out the integument. It can't swallow. This group of mites cannot actually swallow a nematode. Okay, so I'm going to show you a few studies that we've done here and how basically this supports the concept of conservation biological control. And I'm going to lead you towards the idea that, yes, how eventually the idea of how this pertains to using the soil amendment for enhancing plant protection and soil health. So I'm going to start with a study that we did on providing predatory mites with alternative food and how this would affect its ability to control a pest. And here there's really a case study, and we used the housefly as our model organism. And this was done in Brazil with a very strong group on soil mites. And this was done while I was on sabbatical in 2017. And so first, I want to share with you how they reared these free-living nematodes. What they did was they took beans, put them into soil that was grazing land, and then they came back after a few days, and they collected these beans, and the beans were covered with free-living nematodes. And it was a nematode that's actually, it's phoretic on flies, and this is a free-living nematode that feeds on bacteria. And so they cultured this free-living nematode in this, we called it this bean soup. And basically it was a very rich diet. So it contained bacteria and one-celled organisms. And we also analyzed the bacteria and found that this contained plant growth promoting bacteria, and also bacteria that were toxic, that produced toxins to root knot nematodes. So it was really a complete jungle, and they created this just by collected this community of organisms, nematodes and bacteria, just by placing an amendment basically into the soil. And here you can see this is the predator mite that we used in the study. It's a genus, Macrocellus, that's associated with flies. It attaches itself to the fly, the adult, and it moves with the fly from one place to the other. And it feeds on both the fly eggs, the fly first instar, and it also feeds on nematodes. And the nematodes also travel with the fly. So here, you see the predatory mite moving its chelicera and basically feeding on the nematode as if it's a piece of spaghetti. So in this study, we started out looking at small arenas, and our question was, if we provide the fly with additional food, or if we provide the predator, Smacrochellus ambersoni, with a pest and additional food, or if we provide it only with a pest, where will we have better control? And actually, what happens is when you look at only a very small system, sort of like a Petri dish, if you add alternative food, you actually lower the effective biological control. So this is the amount of L1 larvae that were fed upon. So you see more L1 larvae of the fly were fed upon when you don't provide alternative food. So providing alternative food lowers the prediction. But then we went another question we wanted to ask, that we asked in this system, in this very controlled system, was, and this again will pertain later on to the idea of using black soldier fly as an amendment, is what happens if you feed so what happens when you feed the nematode with a very with a diverse diet versus feeding the nematode only with one food source. For nematodes, it doesn't matter what you feed. They can feed on a diverse diet, they can feed on yeast, and in both cases, they do very well. They develop into high numbers. But we wanted to ask, what effect does the food that the nematode eat have on the fecundity so the fecundity is how many eggs the predator lays per day on the predatory mite. So here we fed the nematode only yeast, and here we fed the nematode this zoo, this very diverse diet, this bean soup. And you can see that the fecundity of the predatory mite was much higher when it fed on nematodes that fed on a diverse diet. So again, the idea of what effect you can have by making the system more diverse. And here you're looking at, we changed the spatial scale of the experimental setup and also the temporal scale. So how long the experiment went on and how large the arena was. And here we provided the predator with only fly eggs in the diet that flies develop versus fly eggs and the free-living nematode. And here you can see that, so the green line is when we did not provide nematodes, and the blue line is when we provided nematodes. And the parameter that we evaluated to determine the level of biological control was fly emergence. So how many flies emerged from the time that we added the eggs to the container after basically the predators could feed on the eggs and on the larvae, and in the end, the more they fed on these immature stages, the less fly emergence would be. So in the first week of fly emergence, actually, we saw the same result. So the more food they have, the less biological control we had. So the more fly emergence we had. So here, the more food they had, so we had both flies and nematodes, the higher the fly emergence in comparison to no alternative food. But in the second week, you see the opposite happens. Our fly emergence continues to go up where we did not provide alternative food, and where we did provide alternative food, the fly emergence went down dramatically. And here is the number of predators we found in the containers at the end of the experiment. So here we found 430 predators, and here we found more than 1,100 predators. So almost 4 times the amount of predators when we added alternative food. So when there's more food in the system, the populations of the predators are higher, and the biological control is better. So now moving on to a second study that we did in this sabbatical that I did, and here it's the data that I'm showing is both from Brazil and also from the time that I worked at the USDA, and here we looked at more agricultural systems. So we looked at tomato plants, dwarf tomato plants in pots, and we used the same free living nematode as alternative food. And the idea was we wanted to see how this supplement of nematodes in their culture media, how this would affect root knot nematode control and plant health. And so again, we use the same, this is a different predatory mite, Stratiolipes schimatus. And it's the same order, Mesostigmata, but a different family. And here you can see, again, the predator. You can see between the predators, palps, the nematode. And here you can see the different appendages of the mouth parts colored. So this is the front leg that senses the nematode. These are the palps that are holding the nematode. The nematode is colored in blue here, and these are the corniculi. And at the end, you can see a hole. This is where the saliva is excreted from, and where you can see the predator is about to spear the nematode and inject the saliva into the nematode, which allows for extraoral digestion, and then the mite macerates the nematode between its chelicera, and here you can see this yellow appendage, which is the labrum, which basically allows the flow of the liquid to enter the mouth. And here you can see the plant parasitic nematode, the root knot nematode, its only motile stage with its stylet, which it uses to pierce and enter the plant's roots. This is the only stage that's motile in this organism. So we had on this project, we had postdocs and researchers. This is the tomato research group and a plant nematologist, plant parasitic nematologist, a bacteriologist, an entomologist with bacteriologist. We had postdocs and bachelor students. So we looked at how we could so we had 8 treatments with or without free-living nematodes in their culture media, with or without predators, with or without the root knot nematode. And we ran 2 experiments, one for 5 weeks and one for 15 weeks. And we had a very interesting result in both experiments regarding the effect on root knot nematodes. And so the indicator of what level of control we had was the number of galls of root knot nematodes. And here, the left columns of the figure are with free-living nematodes in their culture media, and the right columns are without free-living nematodes. And here it's with predators and without predators. So you can see that when we provided free-living nematodes in their culture media with predators, we had the lowest number of galls, both after 5 weeks and after 15 weeks. And when we had without free-living nematodes and without predators, we had the highest number of galls. What was really interesting was that where we did not provide predators, or when we provided predators and we did not provide free-living nematodes, the result was the same. So with free-living nematodes in their culture media without predators, or without free-living nematodes with their culture media with predators, the level of galls was the same. So this is an indication of there are different mechanisms going on here. We also found, as I said, in this culture media, there were plant growth promoting bacteria. There were also bacteria that produce toxins that are toxic to root knot nematodes. And so, so when you're looking at it in a holistic way, or your approach is holistic, you're adding, yes, nematodes in the culture media and predators were having different trophic levels and different compounds and different bacteria were actually operating on different mechanisms that in the end provide varying levels of control of the pest. Free-living nematodes are very important for different reasons in our soils. They contribute to nitrogen mineralization by feeding on by releasing the nitrogen in the bacteria. They feed on bacteria. They move bacteria from place to place because the bacteria attach themselves to the integument of the nematode. They they're moved in two ways. One, as I said, they attach themselves to the integument. Two, only a proportion of bacteria that are fed upon by the nematodes are digested. A large proportion of the nematodes just go through the gut of the nematode and are secreted live from the nematode. And so the movement of the bacteria within the soil is very important. So this contribution of movement is an important part. So what you see here is what effect the free-living nematodes had on the amount of nutrients in the plant. So here we can see that when we provided free-living nematodes, we had, if we look at the potassium. So without here are the root knot nematodes. Yes, they impair the ability of the plants for nutrient uptake. So this effect of free-living nematodes was only apparent when you consider only plants that were not infested with root knot nematodes. Here we had a higher potassium uptake when we had free-living nematodes. And here, so this is, in other words, we're comparing this A to B, and the same thing is true for nitrogen. We compare without root nematodes with free-living nematodes versus without root knot nematodes without free-living nematodes. You can see that there's a higher uptake of nitrogen and a higher uptake of potassium. Okay, so as acarologists, we need to know, in order to take advantage of this biodiversity, we need to know, and we need to identify these mites, and this is not a simple ordeal. And what we've done is we've tried to work in multidisciplinary teams where we combine molecular studies and morphological studies, so that in the end we produce barcodes that the community on the whole can use these to take advantage of these biocontrol agents. And these are examples of publications that we've done combining these tools. So in the end, the ecological community can collect these mites, basically squash them, barcode them, and then with barcodes that are added by studies such as these, people will be able to use these organisms and take advantage of them. So, how does conservation biological control connect to conservation agriculture? And if people here are from the regenerative farming community, so these principles are obviously known to them. So we want to minimize soil disturbance. We want to keep a permanent crop cover, and we want to diversify by crop rotation and by adding cover crops and so on. These principles are very important for conservation agriculture, and the use of frass as a biofertilizer basically is part of this approach, and it really joins and links between conservation agriculture and conservation biological control. So so the last section of my talk will be on harnessing the food web, harnessing the soil food web for the biological control of root knot nematodes and for soil and plant health. What I'm going to show you is part of a research project that is going on for more than 2 years, and this was a project that was submitted to the scientific foundation, or the science foundation of Germany, the DFG. It's a trilateral project, between Germany, Israel, and Palestinian authorities. And so these are the images that I showed you before: the feeding on the root knot nematode and the feeding on the free-living nematode. So again, what is the idea of it feeding on the free-living nematode is that these predators will be abundant in the soil, and they'll be able to control the root knot nematode before it causes damage. So our general hypothesis is the right soil amendment, and here we'll call it also biofertilizer, can conserve and rehabilitate beneficial biota for root knot nematode control. Okay, so the people from the Black Soldier Fly Consortium, I don't have to convince, but I'm happy that there are other people here that actually are not really familiar with the Black Soldier Fly, and certainly not its frass. So why Black Soldier Fly, and why Black Soldier Fly frass as a biofertilizer and soil amendment? So recent and current studies indicate it reduces root knot nematode damage. It contains chitin known to induce plant resistance to pathogens. A high proportion of the microbiota in the black soldier fly originates from the black soldier fly gut, which provides a foundation for repeatable results. So this is very important because growers, when you speak to them about using frass as a soil amendment, so they ask, so how is frass different from compost? And there is a very big difference because compost is obviously it's organic matter, but during the composting process, first you kill all the bacteria or most of the bacteria in the compost heap, and then the compost heap is recolonized by bacteria. And you don't really know with what bacteria this recolonization, in what bacteria you'll have in this recolonization, whereas with the black soldier fly frass, even with the heating that is now required by the European community for 1 hour, you still have a bacterial community remaining, and an important proportion of this bacterial community originates from the black soldier fly gut. Now we started this project and wanted to see whether the black soldier fly, whether we could rear free-living nematodes on black soldier fly frass, and we were very successful in that. And of course, this industry is growing as we speak, and there will be substantial amounts of frass that can be used for that will be able to be used for soil amendments. So biofertilizer can be defined as biological products containing living microorganisms that, when applied to seed, plant surfaces, or soil, promote growth by several mechanisms such as increasing supply of nutrients, increasing root biomass or root area, and increasing nutrient uptake capacity of the plant. I think it's really important that we understand that that the frass that with a frass, we can be manipulating, changing the bacterial community of the soil. So it's not only adding bacteria, it's diversifying bacteria, and this bacteria could be dependent on what the black soldier fly feed on. So it's really this idea of living, a biological product containing living microorganisms is very important. So I'm going to show you results from two experimental setups, one from potted plants and the other from greenhouse. And in the potted plants, we wanted a natural infestation of root knot nematodes. And so we were in touch with growers and who claimed that they had root knot nematode problems. When they finished their cropping cycle, we went and collected soil. We first verified that the plants had galls, and then this soil will be used as our potting mix, along with or without frass, and with perlite to aerate the soil. And in this potting plant experiment that went on for 6 weeks, we determined at the end of the experiment the level of root knot nematode galls, the biomass, and the abundance of free-living nematodes and mites. And ongoing, I don't have results to show you now, but also we determined the abundance and biodiversity of the microbiota. So this is the effect on biomass. And you can see that, so here, so we did this study in 4 sites, and in each site we had a field site and a greenhouse site. And you can see that in all the sites, we had an effect on the plant biomass, at least a numeric effect. And this was true for all the field, all the field sites. You can see all the field sites were significantly different, and part of the greenhouse sites that were. So in all the sites there was a numeric difference, and in some of the sites there was in some of the greenhouse sites there was also a significant difference. So this was the effect on plant nutrients. We did not see an effect on phosphorus or potassium, but we did see an effect on nitrogen, and also a certain effect on CN ratio. And here, this was the effect on the abundance of nematodes and mites. So the nematodes are on the left side, free-living nematodes per kilo of soil, and this is the predatory mites per kilo of soil. And you can see that the scale is different by 2 orders of magnitude. So this is 7,000 versus 70. So 100 times more nematodes than mites. And we evaluated this 10 days into the experiment, and then at the end of the experiment. The variation 10 days into the experiment was quite high. You can see that there were far more nematodes with the BSF than without. But the variation was high, so this wasn't significant, but this was very significant. So at the end of the experiment, we had 20 times more nematodes where we added BSF than where we didn't. And there was the same relationship with mites, but the mites, there were far fewer, and the variation was very high. So basically, here the addition of free-living nematodes helped to conserve the populations of free-living nematodes. And this is now, what do these free-living nematodes eat? And most of the nematodes that were found here were bacteriivores. So basically, this is an indication that there was far higher levels of bacteria with the BSF than without the BSF, because this is what they eat, these nematodes. So this result is, so I don't have the microbiota abundance here yet to show you, but this basically is a clear indicator that we changed substantially the microbial abundance of the potted plant only by adding 5% frass by volume. So this is really a strong impact on the bacterial community. And this was a really quite incredible result. This is in 2 sites. We had a significant effect on the level of root knot nematode abundance. Basically, it was the same result that we had in our potted plant tomato trials, where we had far fewer galls. So this is with the BSF, without the BSF, and this is in the greenhouse, an organic greenhouse. So we significantly reduced the root knot nematode abundance on these plants. Now, this is a regulation that is now in force in the European community. In other words, people who want to use frass need to heat the frass for 1 hour to 70 degrees. And this was decided because because insects, the black silver fly and insects in general can feed on anything, including manure or whatever. And in order to avoid infecting the soil with a harmful bacteria, this is the regulation that they've enforced. Okay, so just to summarize our potted plant trials, using spent soil, foliage dry weight was enhanced, foliar nitrogen increased, and so did this the CN ratio, but no effect on potassium and phosphorus. Both free living nematodes and predatory mites decreased from day 10 to 60, but abundance was higher with the frass application. And okay, this has to do with, I'm not going to go into this, these are different functional groups of nematodes. I haven't shown you the data, so I won't bother with this. And the root knot nematode gall abundance was lower on plants with the frass application. And just this was here we wanted to see whether pasteurization would affect the would have had an effect on whether the enhancement of biomass. And it was also a study or an experiment that we want to conduct before we went to the grower. We wanted to make sure that frass at different different levels would not have a negative impact on the plant. So we used frass that was pasteurized versus frass that was heated to 7 degrees versus frass that was not. This frass was frass that was not fresh. It was several months old, and actually we didn't really have an effect on biomass. Very limited effect. The effect was better without the pasteurization than with, but in all, I would say this. Basically, we did this experiment just to make sure before we go to the grower that we're not going to have a negative impact on his crop. So we said, okay, we can go ahead. But in retrospect, these results were really not very indicative of the potential of frass, and we'll soon see why. So I'm now moving to the last set of results that I'm going to present, and this was at a commercial. This was basically a commercial scale. It was an organic greenhouse, and I want to acknowledge Danny Pluda, who's the grower of this. It was the grower. These greenhouses are his, and he basically Danny Pluda and his wife are the growers here, and Daniela. No, not Daniela. Sorry, and Anyways, these are spectacular growers, and they really were fantastic hosts. And I think it's because they're such good growers that that this experiment really succeeded. So what did we do? We evaluated different levels of frass, so 0, 2.5, 5 heated versus 10%. We applied the frass before we began the cropping cycle, and then a week later, and then we started irrigating, and then a week later we added the grower planted the crop, and we also evaluated the evaporation of ammonium. And these is the different levels. So we had 2 days after the application of the frass, we saw different levels of ammonium that was trapped in ammonia traps. Basically, we had a correlation between the amount of frass applied and the ammonium released. But this was significant after 2 days, and after 3 days, it was already not significant. And we planted after 6 days. This is the linear irrigation between these levels of frass. This is how we evaluated leaf area, and you can see that the higher the level of frass, at least for leaf area, the higher the leaf area. So we had actually a linear regression between frass that we added, between the percentage of frass applied and the leaf area, except for the 5 heated. You can see the 5 heated actually wasn't different from the control. And this is yield. So this is the number of cucumbers that were picked after 70 days. The way we did this was we counted the number of fruit stems that were severed, and this was basically the number of fruit that were picked. And then we multiplied that by the average weight of each fruit. And here you see basically yield in kilos per plant, and you can see basically the number or the yield per plant was really very significantly affected by all the concentrations that we evaluated, except for including the 5 heated. But you can see that the increase in yield was substantially higher when it was not heated than it was. And this could be an indication of that we killed part of the beneficial bacteria, but it also could be an indication of whether the ammonium concentrations were differently affected in the heating. We don't know, but certainly the heating has a negative impact on yield enhancement. And this is the enhancement of yield per concentration. So per concentration, with its adjacent control, this is how the yield was enhanced. So we had a 41% yield, or roughly 40% yield increase with 5 and 10% frass. At the same day that we did this yield enhancement, we evaluated the nutritional content of nitrogen, phosphorus, and potassium, and we did not see an effect. There was an effect on biomass, but the variation was very high in the end. The only significance we could really show was with and without frass, there was a significant effect, but the variation was very high. So that's basically what I wanted to share with you. I think we have a lot of work in front of us. There are, as I indicated, we are working with other scientists, additional scientists today, and I just came back from a sabbatical in Portugal, and they will be evaluating the organic compounds in the soil water that we've extracted. They will be extracting DNA from the soil and this will be sequenced and subsequently evaluated by Sherry Freilich. We are looking with our German group. We are looking at how these how this frass has affected the fatty acid compounds, the fatty acid abundance in the soil, and how this can be related to nematode abundance and mite abundance. So really, now I've showed you the impact on yield, on foliage, on biomass, but basically we're working now on what mechanisms can be involved here that are actually that are responsible for this really quite spectacular result. And with that, I'm happy to accept questions.
So, Eric, thank you very, very much for this fascinating talk. And yes, this part of our seminar today will be open for questions. And I've seen already in the chat a few, so please go ahead and just ask. And Eric, we have plenty of time for full answers. So thank you.
Eric, I have a question. I am familiar with the work of Moritz Gold from ETH and his PhD. I think they have very similar data on the effect of pasteurization of frass, and they were able to attribute the effect of the beneficial effect of not treated frass to the presence of different fungi in the soil, beneficial fungi. Are you familiar with that? I mean, can complement your knowledge to some degree.
So one, I'd be happy to see his PhD. I'm not familiar with that. But we will be looking at this. So we are looking at the, we will be analyzing the bacteria and fungi in the soil with and without the frass, or with and without frass at the different levels. So this is still in front of us. So we will definitely be looking at this. And we'll also, as I said, be looking at the fatty acids. And these fatty acids can be connected with, in groups, they can be connected to different microbiota in the soil. And we'll also be looking at the nematode abundance, which feed on these fatty acids, and so we will have more information whether really this involves bacteria and fungi or both. Yeah.
Have you evaluated different diets? Because I think there will be a major impact. And, there's a lot of works about the relation between the diet and the microbiome of the BSF.
Yeah. Okay, so the potted plant experiment I showed was with a diet that Lior has been working on. So it's with beer and apple waste, and the greenhouse experiment was done with the BSF from Friesen, which was from chickpea, as you know. So these are two different diets. Both had similar effects on nematodes, on nematode abundance. So we'll see. But I agree that the black soldier fly diet is really very important, and I think in the end it will also relieve us of this regulation, or at least modify the regulation or the implementation of this regulation. Because if we can define what diet the black soldier fly is fed upon, and if we can define certain waste cycles and identify what bacteria are in these waste cycles, then we can, I think, approach the European community and say, here, we're losing very important effects, nutritional effects of the black soldier fly frass for really no other reason, because the diet of the black soldier fly does not contain these harmful bacteria. So I think it's really a matter of, once this industry gets rolling, and there'll be certain streams of waste that can be identified and standardized, then I think we'll be able to do without this this requirement of heating.
Yeah, I will be I will be willing to help because I've been in touch with the General Secretary of IPIF and I know the requirements. So we can just gather, a very nice package of results and, present them. Yeah. We can coordinate the action if you want. Yeah, I agree that there is a great potential.
Yeah, and we're losing this potential because of, and also this, this heating for 1 hour is really problematic. What volume?
Yeah, I know, but this is due to the regulatory position and, the attempt to categorize frass as a manure. And this is how we treat manure. Okay, so I know that IPF right now are trying to establish the heating for 1 hour as an endpoint, as a valid endpoint. It's just a regulatory step to approve frass for commercial use, because frass is not approved as a soil amendment as of yet. So this is the first step.
Okay.
Thank you, Julia. And anybody else has questions? Because I've seen some in the chat. So if Yoram or anyone else would like to ask, it's your opportunity.
Yeah, going back to the frass heating issue, if I may, I wonder when we feed, is a question to be common, when we feed the BSF basically dead animals, what is known in Hebrew as psadim, not sure the English term for it. We obviously were much more worried about pathogens, salmonella, pathogenic E. coli, whatever. And then heating the frass doesn't make a lot of sense. Obviously, with the cost that you lose a lot of its quality, but the balance is tricky here, right?
Yeah, I agree. So it really depends on the way you're saying. It depends on the waste, but if black soldier fly is going to be used in circular economy, then you'll be able to identify certain streams of waste that this regulation makes sense, and others that don't.
So you're thinking again, it's a regulation issue more than anything else. If you feed certain streams, you need to heat. If not, you don't.
Yeah, I think that in the end will yeah.
Thanks. Makes sense.
Gal, do you have a question?
I have a question.
Yeah.
Hi, Eric. I have a question. Did you ever try to see the difference in the microbiome of the soil when you use sterilized or so-called pasteurized frass versus non-treated frass?
So we're really this is in the pipeline, Zev, and we should have this information actually very soon. So it's now it's about to be, the DNA is about to be extracted and sequenced. So probably in a couple of months we'll have the data. But everything is it's in the pipeline.
Gal, do you have a question?
No, it's okay.
Okay, anybody else had something to ask?
I have another question, whether the heat treatment changes the bacteria profile in the frass versus the non-treated. Because it might be that you kill most of the bacteria. Or let me rephrase it. I assume that the frass is rich with bacteria that are resistant to chitin. Chitin has an antibacterial effect, and maybe that is the first selection. And then when you heat treat it and you kill most or part of the population, that causes the effect. And the question is, if you could theoretically reconstitute the bacterial population with a treated frass, would you gain the same activity like non-treated frass?
Well, actually, in this result, so this was effect of the different frasses and with heating and without heating on leaf area. And you can see that there was no significant effect. There was no difference from the control, okay? Whereas with time, so this was done 3 weeks after planting. And this was done 70 days, so 10 weeks after planting. And here we can see that it looked like the frass that was heated time did have like a recuperating effect on the positive effect of the frass, because you can see that there's of course, so if we compare the 5 heated versus the 5 not heated, okay, the impact was much better without the heat, but with the heat, 10 weeks after planting, there was a significant effect, a very significant effect on yield. So it's very possible that the communities were significantly reduced, but with time, this situation improved. So there was a question from Sharon in our deliberations on how to consider frass and how many times it should be added. And so in this experiment, we only added it once before planting.
Eric?
And I think that basically we're changing the community, even with the heating, we are changing the abundance and probably the community composition. Well, but this we'll know soon. All this we don't have to guess here, because soon we'll have data on this, on this question, whether are we only affecting abundance or are we affecting also the biodiversity in the soil.
Eric, can I ask a question about this? So in this experiment, did you consider doing as a control treatment with an NPK, with a NPK fertilizer that mimics just the NPK of the frass to differentiate between the two effects, the effects of the microbiome in the community and the effect of just the NPK that comes from the frass, which is expected to increase yields?
Yeah, so thanks for your question. So first of all, in this experiment, in the greenhouse experiment, so in the potted plant experiment, we actually worked on spent soils. There was no fertilizer. Yeah, and there we found this effect. Yeah, that in all the experiments basically had the enhanced biomass. And what you said is really relevant. You know, basically we added a fertilizer here, and so we could have a fertilizing effect. But when you look at the effect on nematodes, then obviously you're not having an effect on fertilizer, you're having an effect on bacteria. But in this trial, we added the frass on top of the fertilizer. So here in this organic grower, he finished his preparation of the soil, and on top of after the greenhouse is ready for planting, we added the frass. So basically, all the nutrients that the grower was interested in adding with his compost, he added, and here we added on top of that. And okay, that's one thing to take into consideration. To further respond to your question, so with Leora, we now have a chief scientist project where we are evaluating exactly what you asked. So we have a treatment. We have a controlled treatment without fertilizer and without frass, and we have a controlled treatment with NPK, and we have on top of that we have frasses that were heated, not heated, heated at 50 degrees, heated at 40 degrees, so on. So we will have that. We will have more information on that.
Thank you.
Are there any other questions, or can we finish this seminar? Anyone else? Last chance. Okay, so Eric, I'd like to thank you again.
Like before, I just wanted to say I hope it was okay that I ended with the frass. I really wanted to give. I'm not I'm not a water and soils person. I'm really an agroecologist and a biocontrol person, a person who believes in conservation agriculture and conservation biological control. And really, what I tried to give you, and I don't know how many people are left here. If it's only the 5 that I see, I hope that I didn't tire everyone out and they just left. But basically, what was important for me was to give you, and it was an opportunity to give you this more holistic approach. Why I believe frass is really an important soil amendment, and why soil amendments are important for conservation biological control, conservation agriculture, and regenerative agriculture. Thanks.
Well, I think you did an excellent job, and yeah, it was fascinating. And Sharon, would you like to say something for the end?
Yeah, well, thank you very much, Eric, for your time and for your presentation and for sharing your knowledge internally with the BSF Israel community. Thank you all for participating. We had, by the way, Eric, a solid 56 people attending, some going up and down, dropping, but that was most of the time. So I think your topic was of interest. Whoever wants to continue listening to our program of seminars, we'll be having a seminar probably every couple of months, different areas around the black soldier fly. Please join our LinkedIn page, and we will be publishing our future seminars there. Thank you very much for everybody. Thank you very much to the Israel Innovation Authority for supporting this program. And I hope to see you next time. Bye.
Bye.
Thank you.
Bye.
Thank you, Eric. Bye.
Thank you. Bye.
Bye.